Our today's topic is reproduction and economic importance of Ephedra. Ephedra is dioecious with male and female cones occurring on separate plants. Only Ephedra foliata is monoecious. The male and female strobili are borne in the axil of leaves of the determinate shoots and also in whorls on the nodes of older branches. They may be solitary but mostly arranged in dichasial cymes. Dichasial cymes is a type of cymous inflorescence where the main stem stops growing and produces a flower, and then two lateral branches emerge from the base of that flower, and each also terminating in a flower. And this branching pattern continues with each lateral branch giving rise to two more. In Ephedra, flowering usually takes place in March through May, and seeds ripen from June through September, depending on elevation and species. Plants are wind pollinated. Ephedra plants do not flower every year. Their reproductive pattern could be described as massed fruiting, where most individuals in the population flower synchronously in a year with ample rainfall and large quantities of seeds are produced. The population does not flower again for several years, whether or not a high rainfall year occurs. The distribution of male and female ephedra plant is not random. Individuals on dry slopes are overrepresented by males, whereas those growing on run-on surface are four times as likely to be females as males. Next is structure of strobili and gametophytes. So first we'll start with the structure of male strobilus and male gametophyte. Male strobilus or determinate strobili are in whorls of 2, 3 or 4 at the nodes of small green branches. You can see here, look at this first picture. Here this determinate strobili, they are present in whorls of 2, 3 or 4 on the nodes of the small green branches. So this is nodal part of branch, and here these are the strobili. So the male strobilus, just look at one male strobilus. Male strobilus consists of a short axillary shoot bearing 2 to 3 opposite pairs of brackets. So these are pairs of brackets, of which the lower 1 or 2 pairs are sterile, while the rest bear solitary flowers. So in the axil of fertile bracket, just look at this second picture. This is fertile bracket, and in the axil of this fertile bracket is a shoot bearing two thin small opposite means two thin opposite scales. These are two opposite scales, 1 and 2, which are which have been interpreted as variant. So between these scales, the axis is prolonged and bears microsporangia at its tip. So these are microsporangia. Now, structure bearing two scales at the base and sporangia at the top has been called a sporangiophore. So this is sporangiophore. Perianth, this is fertile bracket, perianth, sporangiophore, and microsporangia. In some species, one or more. Usually more sporangia are sessile at the top of the sporangiophore. Now, structure of male gametophyte. As in all seed plants, microspore is the first cell of the male gametophyte. So, at its first division, perithelial cell is cut off by a wall. Just look at this first figure. This is perithelial cell. At the second division, second perithelial nucleus is formed. So this is perithelial cell 1, perithelial cell 2, and at the second division, perithelial nucleus is formed, but there is no wall separating it from the rest of the spore. So rest of the spore, which is the Enthrydium initial. So at this stage, 3 nuclei formed, one perithelial cell 1, perithelial cell 2, and enthrydial initial. Then this enthrydial initial divides. So this a diagram showing division of the enthodial lineal. So the lineal then divides, forming a tube nucleus. Just look at this diagram, tube nucleus and a generative cell. Generative cell divide to form the nuclei of stalk cell. This is stalk nucleus and this is body nucleus. So uh, means when generative cell divide, this part is showing division of generative cell. Two nuclei are formed, stalk nucleus and body nucleus. So these two nuclei, just look at this part, these two nuclei lie in the common mass of cytoplasm and are never separated by cell wall. So at this stage, pollen is shed. When it reaches the ovule, this generative cell divides, giving rise to two nuclei, which are alike in size and appearance. Then exine outer wall cracks longitudinally, and the inner wall, that is entine, it slips out and becoming prolonged into short pollen tube. Next is structure of female strobilus and female gametophyte. So female strobilus or ovulate strobilus, it can be distinguished from the staminate strobilus even in the early stages because it is elongated and pointed, while the staminate strobilus is globular. So ovulate strobili, just like staminate strobilus, they are also present in balls. Just look at this first diagram. This is small green shoot. These are here the nodes on the shoots, and these ovulate strobili, they are also present in whorls of 2, 3 or 4 at the nodes of these small green branches. So when we compare the structure of ovulate strobilus with staminate strobilus, here in ovulate strobilus there are more brackets than in the staminate strobilus, usually 4 or more pairs of sterile ones and the terminal ovules. Just look at this diagram. These are brackets of ovulate strobilus with terminal ovule. So ovule has two integuments, the outer consisting of 4 brackets and inner of 2 brackets. And at maturity, means these 2 brackets, they are quiescent at the base and at maturity splitting at the top. So inner integument at the time of pollination elongates. Just look at this part. Inner integument at the time of pollination elongates immensely, and at its top, a sparkling pollination drop is formed. So this is ovule micropilot tube, and this is pollination drop. Then next is female gametophyte. So megaspore is the first cell of the female gametophytes. Its germination begins, as in all gymnosperms, with a period of simultaneous free nuclear divisions. Free nuclear period lasts about 20 days, right? Just look at this part. This part is showing free nuclear division, and this free nuclear division lasts for 20 days and wall begin to appear at 256 nucleate stage. Then wall formation begins at the outside and rapidly proceed toward the center until the gametophyte is cellular throughout. Even before the gametophyte is completely cellular, there is differentiation into upper reproductive region consisting of large elongated cells and the lower nutritive region with small cells. Later, the lower part is further differentiated into upper storage region and below which the cell act as historium, right? Usually, there are 2 archegonia, occasionally only 1 and rarely 3. So, usually 2 archegonia are present in a female gametophyte. This is a part of female gametophyte showing 2 archegonia and how these archegonia are formed. So, first cell that acts as archegonial initial, right? It cuts off, soon cuts off, and 2 cells are formed: neck cell, neck initial, and this central cell. This neck cell, which divides several times, forming 4 or 5 tides before any anticline appears. As many as 8 tides were observed. Since each divides anticlined into 4 or more cells, there are seldom less than 32 neck cells and often more Some of the divisions are so irregular that neck is not sharply marked off from the surrounding cells. When the nucleus of the central cell divides, now let's just come to the second cell, that is central cell. When the nucleus of this central cell divides, no wall is formed between the ventral canal cell. Means when the central cell divides, cells are formed that are ventral canal cells and then egg. So no wall is formed between the ventral canal nucleus and that of the egg. So ventral canal nucleus may then move down with the egg nucleus or may remain at the top of the egg. So just look at the structure of archegonia. These are neck cells of archegonia, and this is ventral canal nucleus at the top of the egg. So this is egg nucleus. So this whole structure of archegonia. Then next is fertilization, pollination and fertilization. A dense moss of cytoplasm, which is evident below the nucleus of the central cell, becomes denser, surrounds the egg nucleus and extends for a long distance below it as fertilization takes place. At the time of pollination, pollen grains fall on the pollination drop and are drawn down the long micropyle. This is micropyle, long micropyle, and by this time, such a deep pollen chamber. Just look at this part. This is pollen chamber. This pollen chamber has been formed by the breaking down of cells in the upper part of the nucellus. So this is nucellus, and this is pollen chamber. That here the pollen grains fall directly upon the female gametophyte. So this is female gametophyte. So a short, what happened next? A short pollen tube forces its way through the neck of the archegonium and discharges all four of its nuclei into the egg. Now, next structure which is formed is called embryo. So next is embryogeny. So there is a free nuclear period with usually 8 free nuclei which do not sink to the bottom of the egg but are somewhat evenly distributed throughout the protoplasm. Around each of these nuclei, there is organized a cell which is young embryo, right? So young embryo is then formed. Usually from 3 to 5 of the young embryos begin to develop. Then nucleus of the embryo cell divide, but the formation of wall between these two nuclei delayed until the suspensor is formed, until the suspensor tube has been formed. So embryo nucleus pass into the tube followed by the suspensor nucleus, and the wall is formed between them, beginning at the outside and progressing toward the center. So now embryo cell then divides and the embryo then develops, and then a ripened seed is formed. Next is economic importance of ephedra. An alkylate ephedrine is obtained from some species of ephedra, and it is used in preparation of medicines that cure cough, bronchitis, asthma and hay fever. Then tincture of ephedra zeradiana is also used as cardiac and circulatory stimulant. Then decoction of the stem and root of some ephedra species are used to cure the rheumatism, and syphilis. Juice of berry then is used to cure respiratory disorders. Then bourbon tea is brewed from the species of Ephedra in the southwestern United States. Some species are grown as ornamental. Thank you.